Okay, so in chapter 9, we're going to look at controlling microbial growth in the environment. And these are our study objectives, which you can read over on your own. We're going to start with, I'm going to emphasize 3 of the terms on this list. The first 3 terms I want you to know are antisepsis, disinfection, and sterilization. Sterilization would be total destruction of all microorganisms and viruses in or on an object, complete killing, absolute elimination of everything. Disinfection and antisepsis both involve death or destruction of most microorganisms, certainly not a complete removal or destruction of all microorganisms. And the main difference between antisepsis and disinfection is antisepsis is on living tissue, like when you're putting some alcohol on your skin because you're getting ready to get a shot. Or disinfection would be on non-living tissue, like you use a harsh chemical or you use heat to disinfect a tool that you might be going to use in a surgery, but you're trying to disinfect the tool or a surface of a non-living object. We will also mention a little bit later pasteurization as a technique of removing microbes in food products, and not complete removal, but just a reduction of microbes in food products. And then degerming, we're going to talk about that in terms of like when you wash your hands with soap, just washing off microbes is called degerming. So those are the terms that you want to pay special attention to. The antimicrobial agents that we're going to focus on in this chapter are mostly going to be the ones that result in removal or complete destruction of microbes on objects or food. So we won't talk very much about antimicrobial agents that can be used in the human body. So chapter 9 is primarily, as you can tell by the title, control of microbes in the environment. So that's non-living items, objects, surfaces, and so on. All of the agents that we're going to discuss in this chapter fundamentally do one or more of these actions. So there are 4 fundamental actions, and different antimicrobial agents will target one or more of these. So there are agents that can destroy a cell wall, damage a cell wall and cause the cell to burst. There are agents that can affect a cell membrane, which means the cell loses its complete integrity when its membrane is compromised. We are going to look really briefly at a comparison of 2 types of viruses. All viruses have some kind of protein, what we call capsid, made out of proteins, and then some kind of nucleic acid on the inside. If that's pretty much all that the particular virus has, that's called a non-enveloped virus. But some viruses, in addition to the capsid and the nucleic acid, have a membrane surrounding all of it, and that membrane is called the envelope. And so any virus that has the capsid and nucleic acid and an envelope is called an enveloped virus. It turns out that non-enveloped viruses are actually harder to kill because they don't have this fragile structure, this envelope surrounding them. When the envelope is destroyed in an enveloped virus, it completely messes up that virus's ability to infect. And so non-enveloped viruses are actually harder to kill, or harder to, what we would say, inactivate. Now, also, you're probably familiar with proteins. Proteins have 3-dimensional shapes, so anything that denatures proteins will destroy the metabolism and the structure of a cell or a virus. And anything that affects nucleic acids, anything that alters or destroys nucleic acids, can also kill or inactivate cells and viruses. So these are the four things that destroy cell walls, cell membranes, proteins, and nucleic acids. So the antimicrobial agents that we're going to look at in Chapter 9 will have one or more of these effects. There is a kind of a spectrum of susceptibility that microbes have to antimicrobial agents. So like I was saying, envelope viruses are actually fairly easy to kill. They're on the end of the spectrum that's called most susceptible or easier to kill. And on the side of most resistant is things that are harder to kill. Now, you may not know what a prion is, but you do have some familiarity with bacterial endospores. Remember that those are produced by Clostridium, the genus Clostridium, and the genus Bacillus. And you're probably familiar with mycobacteria, because remember, that's the genus of the organism that causes tuberculosis. There's a reason why I keep repeating these things. Make sure you remember those. But so on this end, you have some of those things that you're familiar with. Those are pretty hard to kill. And then on the other end, you have envelope viruses, and you have gram-positive bacteria. An example of gram-positive bacteria would be Staphylococcus, Streptococcus. So in terms of comparison, these things are easier to kill. All right. Now, by using one or more antimicrobial agents, you come up with a strategy. Even if you have something that's hard to kill, you can typically come up with a strategy that can be successful. But things that are very susceptible, you may not have to use more than one kind of agent. Ideally, in a perfect world, an antimicrobial agent should be cheap, should work quickly, it should be able to stay good on the shelf for a long period of time, and it should be capable of controlling microbial growth while being harmless to humans, animals, and objects. And again, this is ideally in a perfect world. In real life, some of the agents don't have all of these characteristics, so there's sometimes there's a compromise in terms of, how expensive an agent is, or how long it stays good on the shelf, or how quickly it works, or how much it affects humans, animals, and objects. So sometimes there are compromises that have to be made, and when you have to consider those things when you're using an antimicrobial. If you're using something that you purchase at the store, you would look on the label for the instructions. Okay, now for any antimicrobial chemical, and that's a specific group of antimicrobials, there are things that can affect how well that antimicrobial chemical works in terms of killing. And on this graph, you want to get used to looking at these graphs, and kind of first thing is to look at what the axes are. In this graph, you have time in minutes, and the scale is regular, 2 minutes for each major division here. And the number of living microbes is given in a log scale. 10 to the 1 equals 1 microbe. 10 to the 0 equals 1 microbe. 10 to the 1 is 10. 10 to the 2 is 100. 1,000, 10,000, 100,000. So this is a log scale of how many microbes can be counted. And the experiment that they are kind of describing in this graph would be, let's say you had some microbes, you spread them on a surface, and then you sprayed it over top of it with some kind of antimicrobial chemical, and you waited. And maybe every 2 minutes you took a swab from that surface, and you used a technique to count how many microbes were in that swab of that sample that you took every 2 minutes. And what you might find is expressed in this graph. The same chemical was used in both experiments. So the same experiment was done twice, but in one case, the room was set to be 45 degrees Celsius. In another case, the room was set to be 20 degrees Celsius, which is colder. And what you are seeing from the data is that when you took the swabs from the surface, if the room was 45 degrees, the number of microbes you counted in those samples went down pretty quickly, probably to a point where you reached, by 4 minutes, you had not very many microbes at all in the swab. When you did the same experiment at 20 degrees, the swabs you took did have a decreasing number of microbes, but you only reached the same endpoint, somewhere around 11 minutes as you did when you did it at 45. So at 20 degrees, when that experiment is run at 20 degrees, you do reach the endpoint at 11 minutes that you reached at 4 minutes when you did the experiment at 45 degrees. So what that's telling you is the same chemical works differently, works more quickly at 45 degrees in killing the microbes, and works more slowly at 20 degrees in killing the microbes. So temperature changes the effectiveness, how quickly it can kill. In a similar way, we could run an experiment at different pH levels, and you would see that certain pHs cause that very steep killing curve, and other pHs cause a slower rate of killing. Just as a kind of a sidebar, I want to point out that among microbes, there are what we call biosafety levels. And the biosafety levels are classifications basically of how dangerous a microbe is. And microbes are classified either as biosafety level 1, 2, 3, or 4. And these are typically, we're talking about pathogens here, things that are dangerous to humans. In our lab, we are working with biosafety level 1 microbes, and there might be 1 or 2 biosafety level 2s. I'm probably the one handling those when we do some demos. And then biosafety level 3, we're not going to use in lab, and biosafety level 4, we're not going to use in lab. In this picture here, you see a woman doing some research in a biosafety level 4 laboratory, in which you have to wear this completely enclosed suit with its own air filtration system, gloves and boots. You have to shower in, shower out. It's very strict. For a biosafety level 3, typically you would see a kind of a cabinet. It's kind of like the hood where we usually have our stock cultures in our lab, but it's completely closed in the front. It's completely glass over in the front, and you have to stick your hands into these holes that have gloves built in. And so you can work inside the hood, but the hood is completely enclosed. That would be a safety cabinet. And so we don't have a safety cabinet in our lab because we don't have any biosafety level 3 items. But just so you're aware, there's different levels of rating for how dangerous something is, and there's different rules for how you need to handle safety in those situations. Okay, this is the end of this first segment, and on the next segment, we will start talking about physical methods of microbial control. And we have quite a few of these that we're going to list out, and you just need to know fundamentally what they are. And then we will have a section on chemical methods of microbial control. But remember, there's a big focus right now in this chapter on surfaces and objects. So control of microbes in the environment, and also food products.